Hello, I am Samuele Ciolini, a PhD candidate at the Luxembourg Institute of Science and Technology, and I am enrolled at the University of Luxembourg. Currently, I am in the Jülich Forschungszentrum, where I carried out my experiment about root dynamics and the varied water supply. I was able to carry out the experiment here thanks to the European Plant Phenotyping Network. Let me show you what my experiment was about.
Hydropatterning is a phenomenon consisting in the preferential growth of lateral roots in wet areas of the soil. The objective of this experiment was to induce this phenomenon by creating conditions of soil moisture heterogeneity in a soil profile divided in 4 layers by Vaseline barriers. This was possible thanks to rhizomes inserted in each layer, which were used to inject water pulses. Furthermore, we wanted to test whether this phenomenon becomes reversible whenever the conditions in soil moisture change, namely, if lateral root growth is interrupted in the previously moist layer and promoted in a newly wetted layer. Zeamais was the species used in the experiment. The experiment consisted in 2 treatments and 1 control, each one of them having 7 replica. The plants were kept in a growth room at 23 degrees and a light-dark period of 10 and 14 hours respectively. Each individual plant was kept in a PVC tube with a LED mounted on top in order to guarantee that each plant received the same photosynthetic active radiation. In the treatments, layer 1 and layer 2 were the layers subjected to water injections. One layer received the first water pulse and the other layer received the second water pulse after the first one was consumed. The controls were kept at 15% volumetric water content in each layer for the entire duration of the experiment. Together with MRI images, daily weigh-ins for calculating a transpiration, shoot length measurement, and leaf number count, the soil water profiler was used to measure soil moisture in each layer and to determine how it varied on a daily basis due to root water uptake. The MRI was used every night to image the root system undestructively. This allowed to observe the root system development and responses to the treatments in terms of root growth. This is an example of 4 2D images of the same root system taken in 4 consecutive MRI measurements. An increase in the growth of the laterals in layer 2 is clearly noticeable. The software NMR routing can be used to generate 3D images of the root system architectures as well. These images can be analyzed for several parameters such as root length, diameter, and biomass. At the end of the experiment, leaf area was measured and the roots were collected destructively from each soil layer. The roots were then scanned for a final measurement in root length. The data collected during the experiment have not been analyzed yet. However, the experiment can be considered successful in a way that the hydraulic isolation between layers was maintained. Also, we were able to observe root growth of laterals in the wetter soil areas. Furthermore, the differences in water uptake rates between layers appear to be consistent with the differences in root abundance between layers. However, the occurrence of the reversal of the hydropatterning phenomenon still has to be demonstrated. With that being said, I want to say thanks to the EPPM for funding this experiment. This experiment was part of my PhD project, which was funded by the Luxembourg National Research Fund. I also want to say thanks to Dr. Damma Vardushkoten, Dr. Robert Koller, and Dr. Daniel Flugfelder for their supervision, advice, and help during my stay in Jülich. Thank you for your attention.